Also, thanks for giving me opportunity to share our team's work. And before moving ahead, an honor for me to share this work in front of Dr. Soyeon Lee and Coster. Actually, one of the moment brought me to this field was in 2008 when I was high school student. Dr. Soyeon Lee came to our school and had a talk about the experience about the space flight, and I believe it shaped my career route. So it's an honor to share this work. And also, I think it would be a little bit nervous. I think most of their audience are background on their engineering or aerospace or satellite, but it's kind of a kind of pure biology. So I think it'd be just helpful to introduce about the field and what the field is aiming and what we uh, preparing for future missions. So today's topic is multi-omic dynamics of human space flight and interplanetary biology. Before moving on, I think it'd be good to introduce about my background as it is directly connected to today's topic. So in a high level, I'm working on the precision medicine, and as a tool, I'm using the molecular biologies to understand the behavior of molecule in biological phenomena. Also, we're working on the genomics, so a study of the DNA and DNA code and DNA structure. And these days, we are actively utilizing the computational tool to understand the biological information written in different types of molecule. So specifically, I'm using two major tools. One is the multi-omics as decoding the language of the life. So omics is a kind of term encompassing the study of the molecule in a holistic way. So in the cell, there are multiple layers of the molecules, such as DNA, RNA, protein, metabolite, or epigenome. So for example, genomics is a holistic study of the whole the DNA, and transcriptomics is the analyzing all of the RNA at the same time. So with the different layer, we can understand the biological underpinning of the biological phenomena. In addition, these days we are doing multi-omics, which is the connecting different layer of the molecules, the molecular omics layers to understand the molecular underpinning, or for example, if there's different types of disease, we could use that one to subtype of the disease or find the biomarker for example, we can have, if one of the patient has this biomarker, we can use this specific therapy. So the multiomics is basically a tool to decode the biological phenomena. And the opposite way, there is a genome reprogramming tools based on the multi-omic understanding. We can now rewrite the biological biology. So for example, we can remove the disease-causing sequence from the DNA, or we can replace that one with a healthy one, or we can use those tools to validate some of the idea. So let me introduce about my journey into the biology and space based on these tools. So I think over the generation and over the humanity, there are continued question for the where do we come from and what are we and where are we going? And for me, it was the stars in the sky and see the light was the two things directly linked to these questions. So for example, seeing the sky, seeing the stars in the sky is a kind of connected, is a kind of point of view inside to out, and is connected to ask why we come from and where we are going. And when we see the light or cell, it's a kind of point of view of the outside to inside to ask what we are and how the cosmos shapes life. So I believe the modern science coming from to answer those questions based on the cosmos and the life. And in the modern scientific revolution arises around the 16th to 17th century based on the two tools, the microscope, the telescope and the microscope, which directly makes available to see the cosmos and then make available to see the cells or life. And this era, I think the matching tools would be the rocket for the telescope and the multi-omic technology for the microscope. So basically, these tools are made available to closer to cosmos and go deeper into the life. And for the telescope and microscope mainly deals with the how we see, but with the rocket or multi-omic technology, now we can add another layer, the way life exists. So so based on my interest in the cosmos, the space and the life, and also the background of this era based on the rocket and multiomics, I start to study about the interplanetary biology. It'd be kind of a little bit a broad term, but basically I'm trying to enable a multiplanetary humanity based on the understanding of the biology and the space. So our team published a perspective paper called A Second Space Age, spanning omics platforms and medicine across orbit. So I'm not going to the detail, but we see there is a qualitative and quantitative changes around 2020 after the huge advancement in the rocket technology and also the biotechnology, representatively involvement of the commercial space sector and the new genomics technology. So based on this background, our team started the Space Omics and Medical Atlas project, which studies about the molecular, cellular, and physiological effect of spaceflight on humans to advance our understanding to make space accessible to all of humanity and support the transformation of humanity into a multiplanetary species. So the concept and terminology recently emerges, so it'll be a little bit make some confusion. So let me introduce briefly about this kind of similar terminology. But there are terminology about the space biology, space medicine, and astrobiology. The space biology mainly deals with how life works in space. And space medicine is focused on the humans, how to protect and treat humans for spaceflight. And astrobiology is more kind of broader term in time and space, so how life begins, evolves, and might exist beyond Earth. For the expedited space exploration, I think I can start from this figure. So x-axis represent the years, and y-axis represent the number of objects launched into space. So the first event would be 1957, the Sputnik satellite from Soviet Union. And after the World War II, there is a space race between the US and Soviet Union. So there were continued activity over kind of 30 years. And after that time, the kind of activity was slowed down. And after around the 2020, there is a rapid increase of the launch, the object launched, the number of objects launched. So it is mainly driven by the huge decrease in the cost of launch to low Earth orbit, driven by SpaceX. And so far, until 2024, when I checked some data, there were 700 astronauts by until the 2024, and the kind of feature would be there is an increased number of the cyan color, which represent the number of commercial astronauts. So after the 2020, there are some commercial astronauts, and it is expected to be grow more and more. So it's not kind of government astronaut with a specific or high fitness. So it could be young or older, or even people with underlying disease could go to the space. So we want to address that kind of question with these changes. And also these days, after the International Space Station, people try to commercialize the space in multiple ways. And also it is the same for the space biology. So there are some space biotech market map. The main three branches would be space health and medicine. So kind of developing a therapy, and second branch would be space bioprocess or food production, or representatively the space crops, and space manufacturing. So utilize the microgravity environment to make a better product. And our team lead and my former colleague founded the BioAstra, and recently I joined the team. So in the BioAstra, what we are doing is that based on the astronaut multi-omics database, we want to provide the solution in space, which could be also adapted in on Earth. So there are, and also we are trying to launch the Twin Astra as a follow-up of the NASA twin study. So we want to send the kind of 2030 monozygotic twin and want to analyze the effect of space flight in the mission. So we will launch this one in next week IAC. So if there's anyone around the IAC in Sydney, yeah, please visit the BioAstra booths. So in the long term, humanity is moving from on Earth to low Earth orbit to Moon and Mars. And currently, the major platform would be the International Space Station, which will be retired in 2030. And also China has their own Tiangong Space Station. And for the orbital launch, SpaceX provides some missions, and Blue Origin support the suborbital flight from the New Shepard program, and there are public flight or satellite lab. And in 5 years, it is supposed to have a multiple commercial LEO destinations, for example, Axiom station or Orbital Leap or West Haven station and Sierra Space Star Lab. And in addition to the space station, it is supposed to have the uncrewed orbital flight from, for example, the Space Rider from ESA and Dream Chaser Sierra Space from Sierra Space. So our team is working with the Sierra Space to send the passive payload supposed to launch next February, but it's kind of it's the first flight from the Sierra Space, so it's kind of uncertain. But we tried to broach some biology experiment through the Sierra Space with the passive payload. Yeah, so also SpaceX has a plan to send the first Starship to Mars in 2026. And I think I can skip this one. So there are some plans for Artemis and Luna Gateway. And after 5 years, it'd be the one of the major thing would be the SpaceX mission. So SpaceX want to send the payload in 2030 with the Optimus robot. And our team, our kind of collaborating team is also touching with the SpaceX business team to send some payload through this 2030 mission. Currently, it's kind of 500 million for 500 kilogram is the kind of lower limit. So we try to gather some big team to support this mission. So in the context of this space exploration era and expansion of the human presence outside of Earth, how do we ready ourselves for the next era and while safeguarding human health and performance in space? That's the question what I want to solve through the biology. And humans did not evolve to survive in such extreme space environment, representatively the space radiation or microgravity. And it has been known that the screen might be a little bit the size of it a little bit small. Yeah, I can't enlarge. So the basic idea would be the rows are the duration of the mission, and the columns are these rows are for the different organ systems such as renal or arrhythmia or CNS or sleep or cancer muscle kind of thing. So as the duration is getting longer and longer, the risk of the health risk is getting severe. So we need to address this problem, especially if we want to send the people to Mars. It would be kind of around 1.5 year space flight at least. So we need to address that one. And temporal changes in human biology in space would be in a minute. The major change would be the fluid shift through the head. So there is a vestibular visual mismatch or space motion sickness. And in 2 days, there would be the diuresis or some plasma volume contraction or cardiovascular set point reset. And in 1 week, there'd be more immune changes, mainly the immune dysfunction or a risk of latent herpes virus reactivation. And in 1 month, the muscle and bone atrophy is getting a rise, and there is a kidney stone risk. And in 6 months, there is a well-known the syndrome called the SANS, the space-associated neuroocular syndrome. So it's kind of syndrome mainly coming in the visual system and nervous system. And as the kind of space radiation, those are cumulative, there might be some risk of the DNA damage, which might cause some mutation. So it's a kind of that kind of representative temporal changes over 6 months. And after return, most of the changes recovered quickly, but some of the molecular changes persist over 3 months or 6 months based on our team's research. So our team has two branches. So one is the SOMA, the Space Omics and Medical Atlas, mainly is a kind of database and the research team, and the CAMBANK, the Cornell Aerospace Medicine Biobank for the astronaut biobank. So we try to share the samples and the data as much as possible to collaborate together and make innovation together. So if there's anyone interested in some biospecimen or the data, please reach out to us. And currently, our team has joined some of the SpaceX commercial flight mission, the Inspiration 4 launched in 2021, and Polaris Dawn launched last year, and the Frame 2 launched this year. And we also collaborating with the Axiom team to collect some of the Axiom crews, and also has a kind of control sets or the parabolic flight sets. So our idea is to collect the sample pre-flight, in-flight, and post-flight longitudinally to analyze the effect of spaceflight on human and whether it is recovered over time. So we collect some blood samples or urine or immune cells and saliva stool and skin swabs. So how our team has started this kind of mission was that the NASA Twin Study. So it was launched in 2015 to 2016, was a 1 year International Space Station flight missions. And our team lead Chris Mason joined this kind of a big collaborate team. And based on this experience, we're building up a new pipelines, and we applied those pipelines to the previous commercial spaceflight missions. So let me briefly introduce about the NASA Twin Study. It was the integrated biomedicine with twin astronaut. So one of the twin went to the space and spent 1 year, was Scott Kelly, and Mark Kelly stayed on Earth, and we collect the sample longitudinally, pre-flight, in-flight, and post-flight, and analyze those samples. Kind of cutting edge technology at the time was kind of 10 years ago, so it's been a while, but at the time was a kind of cutting edge technology. So we analyzed the biochemistry, cognition, epigenomics, gene expression, immune changes, and metabolomics, microbiome, proteome, and physiology, and telomere. So I'll not go into the detail, but the idea is to provide the molecular explanation or molecular mechanism to understand the well-known physiological changes after spaceflight, such as immune dysfunction or some metabolic changes or some mitochondrial dysfunction or cognitive changes. So here are some of the data. I'll not go into the detail, but we found some immune cell gene expression change, which could explain the immune dysfunction or viral reactivation. So, and also did some metabolic profiling. Or for the analyte change, we did some cognition analysis, metabolite, telomere, microbiome, cytokine, and proteome. Mainly the gray area is the duration of the space flight. And the trend is that during the space flight, those aspects are the changes arises, and after return, most of the changes recovered quickly. So we start to understand what's going on in a molecular level, which could support and provide explanation of the physiological changes in astronaut. but still it was a small n problem. There is only one control and only one astronaut. So we trying to find another opportunity, but kind of collecting sample from the government astronaut is a little bit tight and it's not kind of flexible. But there were great opportunity from the Inspiration 4 mission, which was the world first all private mission to orbit. There were 4 crews and led by Commander Jared Isaacman, and it's 3-day space flight in orbit through the Dragon capsule. So we use the same pipeline we developed from the twin study to collect the samples from those 4 astronauts and analyze the samples based on our technology. And the findings from this study has published last year through the Space Omics and Medical Atlas across orbit with collaborate with through the Nature portfolio, and it was a kind of over 100 institution collaboration and over 25 country collaboration. So the major findings is that we try to provide the adaptation to space flight in the human body at anatomical, cellular, and physiological and cognitive levels. And yeah, so there are well-known changes throughout the multiple organs. So what our team did was we provide the database and we collaborate with our collaborators throughout the country to support their idea about the well-known physiological changes. For example, we provide the immune cell change data or some gene expression change data to support the immune dysfunction function idea or support the microbiome shift in space or a space aging idea or brain damage or space anemia or telomere, insulin, skin, kidney stone, list kind of things. So every analysis published it as a separate paper. So it's kind of over 20 papers. So I'll skip the details, but please reach out to me if there is any question on the details. So I'll skip that part for the sake of time. So as I mentioned, we try to share the data and the sample as much as possible to collaborate with each other. So we launched the SOMA data portal, so you can type the gene names here and can see the pattern of those genes in space. And kind of 6 months ago, it's a 6 month ago database, but over kind of 800 people all over the world utilize this SOMA data portal, and there were kind of 2-3 papers published already after launching this portal 1 year ago. But yeah, so we are keep moving on another mission with the same a similar pipeline. So we joined the Axiom 2 mission and published some of the findings as a book, and also joined the Polaris Down. I'll not go into the detail. So we are now analyzing those data to make a kind of another paper package with the other collaborators in the Florida mission. And FRAM2 was launched this year, and one of the characteristics would be we brought the 2 passive payload. It's a kind of box with multiple in a cell culture or tissue culture or microbiome experiment. So here is the video of the FRAM2 launch. So the full astronaut and our 2 passive payload are here on top of the Dragon capsule. So I was able to see this launch at this mission. And yeah, so in the box, we brought the flask tubes to with a multiple experiment. And also SpaceX brought this video in Dragon capsule. So this is the view of so yeah, so they are on orbit, and this is Earth view from the cupola. And yeah, the one of the astronaut take the video by rotating the passive payload. Yeah, so I'll also skip the details. So it's a final part. I can finish it in 3 minutes. So what we can do from the understanding from the biological changes from the omics data. So how can we protect human health using insights from space omics? Our team lead Chris Mason published it the next 500 years book. is a major idea is that we can utilize genome reprogramming tool to make human adapted to Moon, Mars, and for example, if we think about the kind of next 500 years. So one of the approach we are doing is using the CRISPR technology to enhance the radioprotective genes, or it'd be kind of more progressive way, and we are thinking of the utilizing the defense mechanism from the extreme files such as tardigrade. So we have some cell line introducing tardigrade protein into the mammalian cell, and we are analyzing that one to understand the mechanism. And another feature is the mitochondrial stress after space flight, and it persists over kind of 6 months. It's a little bit like a long COVID. So it is a little bit too complicated, but yeah. So the idea is that mitochondria is a kind of metabolic hub of the cell, and kind of a machine generating energy utilizing the oxygen. But when human or any other animal go to the space, the oxygen consumption mechanism has been uh, suppressed, and our idea is that the suppressing the oxygen utilization makes the kind of chain of the molecular changes, which underlies the physiological risk of the astronaut. We start to collaborate with Oscar Pharmaceutical to utilize camphorol to counteract this effect. The camphorol has been found in plant living in the high altitude where there is a hypoxia, so the oxygen concentration is low. So the camphorol has been known to increase the oxygen utilization. So we are using camphorol to counteract the decreased oxygen consumption in space. So kind of one of the validation studies that we use the camphorol in mouse model and organoid model in a radiation perturbation. So for example, the liver weight is decreased after the simulated galactic cosmic ray, but after the treatment with the camphorol, it recovered, and some of the functional markers of the liver function, such as urea production or some of the protein activity, after radiation, the functional marker decreased, but after KMP, the camphorol treatment, it rescued. Same for the high dose. So we we published this study in preprint, and it's under review in Cell now. So hopefully we can publish we can get accepted soon. And yeah, also the final one would be for last year in Milan in 2024 October, I had a kind of a thought, I proposed idea of the kind of a perturbation dictionary to make a virtual cell, and which could be more developed into digital twin in when Dr. Suyan Lee was a judge, and I was selected as a catalyst program. So I'm preparing to actually got the fund with the Human in Space Challenge. Yeah, so we are keep moving with generating more space omics data and develop the countermeasure, and also understand more and more about how the space flight effect on the space. And there are multiple missions we are waiting to join. So SpaceX Fast mission or Virgin Galactic mission, kind of the oldest one would be next year Dream Chaser, and another one would be the SpaceX Starship mission. And yeah, thanks for, I want to acknowledge all the funding and support, and thanks to patient, crews, families, and their resources sample they donate. And thanks for listening, and happy to take any question.